Blood vessels. Blood vessels play a crucial role in blood flow and the delivery of oxygen, nutrients, and other substances throughout the body. Blood vessels are composed of three layers: the inner layer, the middle layer, and the outer layer. The middle layer contains bundles of vascular smooth muscle cells, which play a crucial role in regulating the diameter of blood vessels. These muscle cells contract or relax, directly controlling blood vessel constriction and dilation. Here I'm going to talk about the mechanisms of vascular smooth muscle cell contraction. Let's look at a single vascular smooth muscle cell. Vascular smooth muscle cells depend on calcium influx to initiate contraction. Calcium can enter the cell from the extracellular space to the cytoplasm through the opening of voltage-dependent L-type calcium channels and transient receptor potential cation channels in response to membrane depolarization. Another source of calcium for smooth muscle cell contraction is the intracellular sarcoplasmic reticulum. The intracellular calcium ions bind with calmodulin. The calmodulin calcium complex then joins with and activates myosin light chain kinase, a phosphorylating enzyme which will phosphorylate the myosin light chains on the myosin heads and trigger the myosin-actin cross-bridge cycling. Myosin is composed of two heavy chains at the tail and two light chains at the head. One of the light chains of each myosin head becomes phosphorylated in response to the activation of myosin light chain kinase. Before contraction begins, the myosin heads bind with ATP, and then the myosin-actin cross-bridge detaches. To begin contraction, the ATPase on the myosin cleaves the ATP, leaving ADP plus the phosphate ion bound to the head. In this state, the conformation of the myosin head is such that it extends perpendicularly toward the actin filament, but it is not yet attached to the actin. Then the myosin head binds to the active site of the actin filament. That bond causes conformational change in the myosin head, prompting the head to tilt toward the arm of the cross bridge. This movement causes the power stroke that pulls the actin filament. Once the power stroke occurs, ADP and the phosphate ion detach so that a new molecule of ATP can bind. The binding of new ATP causes detachment of the head from the actin, restarting the cycle. When calcium concentration decreases, due to the pumping of calcium out of the cell, the process is reversed, and myosin phosphatase removes the phosphate from the myosin light chain kinase, the myosin light chain, leading